Okay. Questions, anyone? Okay, for tomorrow, you are going to be in lab inoculating your unknown into, I believe, 4 different media. And you will then see how it grows on those particular media. So to do that, you need a working culture of your unknown. So one of the things you have to do today is you have to pull your tubes out of the incubator, run them through the spec, and see where you get the most dense solution. That is your highest growth. That is your optimal temperature. And then I need to have you make up one slant for a working culture tomorrow and put it in that optimum temperature. It'll grow up overnight fine and give you enough growth for tomorrow. Okay. Additionally, pull out your core plate out of the incubator and stick that into the refrigerator, and we can have you look at that in lab tomorrow as well. Okay? So basically a lot of inoculation tomorrow. Okay? Any questions? Yes, Mackenzie?
The tubes from the optimal temperature experiment, and you just put them under the middle?
Yep, just put them under there. Actually, you know what? Why don't you save your one that grows the best and refrigerate it in case your culture doesn't grow real well, and then you would have a broth culture to use tomorrow in lab. So that would cover our bases a little bit. Okay? All right. Class on Tuesday might have been a little bit confusing. I'm going to attempt to go back and do a little bit of kind of reiteration, kind of reset the context a little bit and describe to you kind of what my goal is. One of my goals in this particular section is to convince you and introduce you to the fact that bacterial classification and taxonomy is kind of weird. And it's not kind of as straightforward and not as clean of a process as traditional plant and animal taxonomy systems that you are somewhat familiar with. I think you get a fair dose of plant taxonomy in botany. I'm not sure how much taxonomy Dr. O'Neill does in zoology, but plant and animal taxonomy has undergone kind of an interesting evolution. I mean, it kind of dates back, we said, to Linnaeus, where Linnaeus decided to develop this system whereby you gave every organism a scientific name, a genus and species name, and then we started grouping things together into large groups, which were subdivided into smaller groups, that became more and more inclusive. Originally, in the Linnaean system of classification, there were 2 kingdoms, plant and animal. Okay? And then over a period of time, as more information was developed, more organisms were discovered, people started to say, these things don't necessarily fit into just these 2 groups. And so I believe it was about in the 1950s, for example, that Whitaker's 5 kingdoms system of classification really took hold. And we now started saying, well, organisms belong to one of these major groups. Plants, animals, fungi, protista, or these monarans, these prokaryotic organisms. Everything else was eukaryotic. And then as more genetic information was gathered and being used more extensively, the 3-domain system of classification is supplanting or replacing the 5-kingdom approach to classification. One of the things that's kind of interesting about taxonomy is that people who are really into taxonomy oftentimes are characterized as being either lumpers or splitters. And the splitters like to break things down into more and more smaller groups. The lumpers say, no, let's make larger groups that are more inclusive. And so if you're a taxonomist, there's that kind of tension between lumpers and splitters. There's another kind of inherent tension in any classification taxonomic system. And that is this idea of practical identification utility, using the system to kind of quickly identify things and make sense of them, versus this idea of the system should also demonstrate evolutionary relationships. And so we have systems that tend to be a mixture of this phonetic, practical orientation. Phonetic comes from the word phenotype, and so a lot of these characteristics are based upon phenotypic characteristics. Phylogenetic classification systems are more evolutionary in nature, and instead of depending just upon phenotype, really ultimately phylogenetic systems really are more about the genes than they are the phenotype. Well, traditional plant and animal taxonomy schemes have done an okay job of kind of balancing these two kind of tensions that are existing in any classification system. Now, let's go to bacterial classification. Okay? The Linnaean system arose in the, I believe it was late 1600s, early 1700s. That's before we really even knew about the existence of bacteria to any extent. Okay? So once we started to discover bacteria, people went, where do they fit into this particular system? And I told you earlier on that bacteria were placed within the plant kingdom, initially the fauna kingdom, and we still see remnants of that when we talk about things like your bacterial, fauna's animals, they're the flora, the plant part. When we talk about the bacterial flora of your body. And so that's kind of a remnant of the fact that in the Linnaean system, bacteria used to be considered to be plants. They were flora. In the five kingdom approach to classification, they found a home that kind of made more sense to them, and that is they were prokaryotes, but all of the prokaryotes were lumped together into one kingdom. In the three-domain system of classification, we've started to understand that there are prokaryotes that are fundamentally different from other prokaryotes. The archaea versus the bacteria. And so in the three-domain system of classification, that's kind of the de rigueur right now, it's the one that's being kind of most widely used and accepted. We've got those three domains, two of which are prokaryotic and one of which is eukaryotic. What you don't see in traditional plant and animal taxonomy systems that is unique about bacterial taxonomic systems is this thing, Bergey's Manual. Okay? Bergey's Manual is kind of the Bible of bacterial classification and taxonomy. And there's no counterpart to it in plant and animal. So what we're doing right now is we're trying to dig through a little bit about what is Bergey's Manual, what's the philosophy of Bergey's Manual, how has Bergey's Manual evolved and changed over time. And one of the things that I'm trying to superimpose on this discussion is particularly this relationship between a practical identification system and a phylogenetic system, because Bergey's Manual has really struggled with this over time. Okay? So you with me so far? Kind of. Okay. So, Bergey's Manual. Let's go back. Maybe one slide. No. There's two types of Bergey's Manual. There is the determinative bacteriology volumes, and there are the systematic bacterial volumes. The determinative bacterial volumes are primarily geared toward being an identification tool. They are primarily phonetic. Although I'll tell you in a minute, in some of these later editions, there was some attempt to kind of squeeze some phylogeny into the Bergey's determinative volumes. There are also then these systematic volumes of Bergey's. There are two editions of this, and these tend to be generally more phylogenetic, although this one right here, the first edition, is still this weird mix of kind of phylogenetic phonetic. Okay? So, Bergey's Manual traces back to 1923. That's when the first edition of determinative bacteriology was published. And every few years they updated this as they discovered more organisms. And up through 1957, the first 7 editions, things were pretty consistent. Okay? This was an identification tool. It was very, very much based upon characteristics of bacteria that were morphological or phenotypic in nature. There was, over a period of time, a little bit more movement from the just basic, cocci, bacillus, gram-positive, gram-negative, to some more physiological and biochemical kind of information that was used as criteria. The organization of these was primarily some keys that were used to identify the organisms. And so that's why we find these early editions, we don't have the first 6 in lab, but the 7th edition, very useful in identification of the species of our unknown. Because the information that you use to key your unknown to species, once you have the genus, is primarily based upon these kind of things we can easily do in lab. Determine a gram reaction, determine cell morphology. Do you have endospores? What kind of foods do they use? What kind of products do they use? So a lot of morphology, biochemistry is kind of the heart of those 7 editions. When the 8th edition was published, things got a little bit different because some of this genetic analysis started to creep in. And the genetic analysis took the form of things like GC content. That's about the best we could do at the time. And that started to kind of inform a little bit how the 8th edition was organized. Because now people were going, we've got actually some tools now that will maybe allow this 8th edition to be a little bit more phylogenetic in nature and not just phonetic in nature. So the 8th edition is kind of a interesting transition, trying to kind of bridge these two worlds. Okay? The first 8 editions, 1 through 8, of Berge's Manual Determinative were broken up into 19 parts. Now, if you're going through Linnaean taxa, kingdom, phylum, class, order, family, genus, species, where's part? There is no part in the Linnaean classification system. This was a totally artificial construct, and it had no taxonomic status. That's one of the ways in which bacterial classification and Berge's Manual is weird. Okay? Because it's got this artificial grouping called parts. What are these parts? A part is really a working group of scientists. And the people who make up these particular working groups that occasionally get together and update Berge's manuals are the experts of organisms in those particular areas. And they got together, each working group got together and said, this is how we're going to organize our particular part of Berge's manual. As you might expect, that can lead to some inconsistency, and it can lead to some differences in philosophy about how things are organized. So that was an interesting strategy. When the systematics were first written, the 19 parts were expanded into 33 sections, but the idea was the same. This was a working group of experts in the area who chose and decided kind of how things were going to be organized within their particular section of Berge's manual. Okay, so 8th edition, 1975. You don't have to remember the dates. Okay. In a period of time between 1984 and 1989, the systematics were written. What were the difference in these? Well, the determinative volumes, there was a single volume for each edition. Now we've got 4 volumes, more organisms to deal with. In systematic bacteriology, we moved from 19 parts to 33 sections. But the big philosophical difference was the shift in this direction from being just a identification tool to being something that was more than that tried to convey information about evolutionary relationships, ecological relationships, okay? So this was a much more kind of ambitious, broad-based way of looking at microorganisms. we'll just take a brief look at this first edition and talk a little bit about kind of what its philosophy was. Once that first edition was in place, people started to kind of go crazy. And they went, okay, you've included all this new information, and it's kind of compromised our ability to use those books as good identification tools. The ecologists love it. The evolutionary biologists loved it because now their kind of information was part of birdies. But this became increasingly a more difficult tool to use for practical identification. So the Birdies Trust went back and said, okay, we hear you. And in 1994, they put together one additional edition, a newest edition, of determinative. And they said, we're going to make a good identification tool for you, an updated one. And how did they do that? They basically pulled all of the identification information out of this first edition of systematics and left out all the ecology, and left out all the evolutionary biology. And so basically they condensed those 4 volumes into an updated 9th edition of Burgess Determinative. Now, for some reason, in that 9th edition, they organized things into 35 groups. Okay? So I just wanted to be aware of when you hear of these things, you know, parts, groups, sections, what do they refer to? There are these working groups that help to organize Birdie's manual. A bunch of experts who say, this is how we're going to organize our particular section. But these particular entities don't have any taxonomic status. They're not a class, they're not a family, etc. Okay? Fast forward to 2001 to 2012, and the 2nd edition of Birdie's Systematic were written. And as I said, we don't have any of those. This is very much phylogenetic and ecological in nature. Its purpose really isn't even to be an identification guide. Why did they decide they could do that? Because they had this 9th edition, which was the most recent identification guide. And so the 9th edition kind of freed up this 2nd edition of systematics to be what everybody wanted it to be. And therefore, it doesn't have this kind of dual purpose, identification and systematics. It didn't serve anybody particularly well. So what we're going to do is we're going to look just at this a little bit right here and kind of see how these evolved into this. And then we're going to spend some time with the 2nd editions to do a survey of microorganisms to introduce you to some of the kind of varied microorganisms that you'll encounter this semester. And we're going to superimpose that on top of the taxonomy of this 2nd edition of systematics. One thing you should know, the 1st edition was based upon a system of classification involving 5 kingdoms. This one now is based upon a classification system of 3 domains. So this one is kind of the most up to date. Okay, so what does systematics look like? Okay, what is his first attempt to make this kind of phylogenetic Burgess manual? Systematics were published in 4 volumes. You see it took them a while to do it. They started in 1984 and finished in 1989. And the 4 volumes of systematics were Volume 1, Gram-negative bacteria of general, medical, or industrial importance. Volume 2, Gram-negative bacteria other than these things called actinomycetes. Volume 3, the archaebacteria, the cyanobacteria, we'll talk about those in a minute, and the remaining gram-negative bacteria. Remaining from what? The ones that don't fit into this criteria, being medically, industrial, or generally important. And then volume 4 are the actinomycetes. And we're going to just briefly mention actinomycetes. So that's these guys right here. We only have 2 of the 4 volumes because anything we deal with in lab is only in volume 1 or volume 2. We're not going to deal with archaebacteria. We're not going to deal with cyanobacteria or those weird gram-negatives, nor will we deal with actinomycetes. Okay? Here's what Berge's manual looks like when you kind of do an overview of it. And it's small, I understand. Okay, so this gives you a little bit. This gives you a little bit more insight into the kind of things that are in Volume 1, the gram-negative, medical, industrial importance, etc. What are in Volume 2, what are in Volume 3, and what are in Volume 4. The numbers in the parentheses over here refer to these sections. Okay? So Volume 1 has sections 1 through 11. Volume 2 has sections 12 through 17. Don't memorize those numbers, okay? So when you start looking through then the 33 sections, what you'll see is that they're organized in a different fashion depending upon the section. Why? Because they had different working groups that organized them. Okay? So, for example, look at volume number 1, these guys right here, sections 1 through 11, and look at section number 1. These are the organisms that we call the spirochetes. Okay? The working group, that is section 1, decided that all of the spirochetes would be grouped into one single order with 2 families and then subdivided into these genera. But then there's this other category down here, other organisms. Okay? This is basically the junk drawer. Okay, this is where you put organisms that didn't really fit into these particular categories. Okay, so things are a little messy. Okay? Section number 2, the aerobic, microaerophilic, modal, helical, vibroid, gram-negative bacteria. It's nothing. Look at the way they've organized. Okay, there's no order, there's no family, there's just a group of genera. Okay, that's how they chose to organize. Okay, look at section number 3. These are the non-modal gram-negative curve bacteria. And they said, well, we'll have a single family with 3 genus, but then there are some other genera. Again, a trunk code, a catch-all category. Section number 4, gram-negative aerobic rods. And they've got things broken down into 3 families with a bunch of genus. So do you see kind of the inconsistency? That's what I really want you to understand. And the fact that people chose to organize things differently depending upon who was in the working group and what their philosophy was. So it's kind of strange, and that's one of the things that I want you to understand, is that Berge's manual can get a little bit strange. What they also attempted to do is they attempted to kind of superimpose some traditional taxonomy onto Berge's manual. And so what they said was that we're going to divide, all of the bacteria in Berge's manual systematics into 4 divisions. And those 4 divisions are the division Bacillicides, those are the gram-negatives, the division Firmicutes, gram-positive bacteria, the division Tenoricides, those are the ones like Bacillus, and then the division Mendocicides, those are the archaebacteria. And so they tried to kind of superimpose some traditional taxonomy on Berge's manual because they were trying to make it more phylogenetic. One of the things that I do want you to understand is that these 4 divisions do not represent the 4 volumes of Berge's manual. So volume number 1, for example, and volume number 3 have members of this division. Volume number 3 has been members of this division as well as members of this division. So again, it's a little weird. And then, where does section come in to all of this? Well, section is kind of superimposed between the traditional criteria or taxa of class and order. So within Bergey's manual, sections are then divided possibly into orders, possibly into families, likely into genus and species. And so this is kind of the taxa that you will typically find within sections of Bergey's manual. And again, section has no taxonomic status, so it's weird. Bergey's Manual of Systematic Bacteriology. Try 2. Okay? This is the one that is now freed of trying to be an identification guide because we've got Bergey's 9th determinative. This is the one that is based upon the 3-domain system of classification rather than the 5-kingdom system of classification. It's expanded to 5 volumes. The previous edition was 4 volumes. What are those 4 volumes? Volume number 1 are the archaebacteria and the deeply branching and phototropic bacteria. Volume number 2 are the proteobacteria. Number 3 are the firmicutes. Volume number 4 is this weird grouping of several different types of microorganisms. I don't expect you to know all of those names. And volume number 5 are the actinobacteria. Where would we find most of the organisms that we would encounter in lab? In volume 2 and in volume 3. This is where pretty much everything we would work with would be found. What I want to do for the rest of today and probably into Monday is give you kind of a little overview of the bacterial world using this organization of Bergey's Manual of Systematic Bacteriology. So basically we're going to take a quick peek through these 5 volumes, pretty much concentrating on the first 3 right here, to kind of give you an idea of the diversity of microorganisms, introduce you to some things you might see again, and probably will see again, and just try to give you some sense of how this fits into this most recent edition of Bergey's Manual, and how it fits into at least what is now the currently accepted classification scheme for microorganisms. So this is a good way to kind of start summarizing Bergey's Manual 5th edition. This slide shows you the 5 volumes, volume 1, 2, 3, 4, 5, and gives you an idea of how it's organized taxonomically. And what we're going to do is kind of pick and choose and talk about some of the representative organisms from some of these particular groups. At the end of this PowerPoint, I will give you a summary slide that says, what do you need to know about classification and bacteria for the first exam? Okay? So it'll give you kind of a summary of what's the take-home message from some of this stuff that we're going to go through. So basically what we're going to do is we're going to go through each one of these volumes, spending some more time than others. Let's kind of look at a big picture first, okay? Number 1, if you look at the first volume. The first volume actually contains organisms from 2 domains, 2 of the 3 domains. The domain archaea and the domain bacteria. Volumes 2, 3, 4, and 5 are exclusively members of the domain bacteria. So this is the only place you find the archaeas in the first volume. Everything else belongs to the domain bacteria. Number 2 point that I want you to see is look at the importance that GC content has in organizing things in Bergey's Manual Systematic 2nd edition. You're put into a different volume of Bergey's Manual depending upon if you have a low GC content as opposed to a high GC content. Okay? Both of those groups of organisms are gram-positive in nature, but something that fundamentally separates them are these have a low GC content, these have a high GC content. This fifth volume is a little bit of kind of the junk drawer again. There's all these weird organisms that are kind of lumped together, in part because we're not quite sure what to do with them. Although the more genetic analysis that we do, we find that there is an evolutionary relationship between those particular organisms. So those are some of the kind of big picture things I want you to see to begin with. So let's take a look at volume 1 first. Oops, yeah. So volume 1, what does it consist of? It consists of two domains, the domain archaea and the domain bacteria. So let's spend a couple of minutes, we've talked about this a little bit already. What are the fundamental differences between the archaeans and the bacteria? Okay. So this particular slide is a good summary of what are the general comparative differences between the three domains: eukarya, archaea, and bacteria. Okay. Cell type. Both of these are prokaryotic. This is eukaryotic. Pretty simple. Okay. Chromosomes. Typically single chromosome in both the archaeans and the bacteria. And typically the chromosome is circular in nature. So these guys are haploid. Okay? So there's not this idea of haploid diploid in the bacteria archaean world. There's just a single chromosome. We have several chromosomes in the eukaryotes, and we have this idea of alleles and homologous chromosomes. Okay, that doesn't exist in that world. Types of ribosomes. Well, archaeans and bacteria both have 70S ribosomes. Eukarya have 80S ribosomes. But the archaean 70S ribosomes have more similarity in structure to the 80S than they really do the 70S bacterial chromosomes. So that's kind of cell membranes. We talked about the fact that there was a little bit of difference in the cell membranes. And actually, eukarya and the bacteria are more similar than the archaeans. These have the ester linkages in their phospholipids. These tend to have ether linkages in their phospholipids. So those archaeans are kind of weird. Okay, they're prokaryotic in many senses, but in some respects they're more eukaryotic, especially biochemically, than other bacteria are. So they really do kind of deserve to be in their own grouping, in their own domain, because they are really quite different. One of the groups of archaeans that's pretty interesting are those archaeans that we refer to as extremophils, and I think I used that term earlier on. And these are organisms that tend to live in fairly hostile environments. Two large groups of the extremophilic archaean bacteria are the halophils and the thermophils. What does thermophil mean? Heat loving. Okay. These live at extreme temperatures, like a boiling hot spring. This is at Yellowstone Park. And the water in those boiling hot springs can reach temperatures of 212 degrees almost Fahrenheit, 100 degrees Celsius. Temperatures which you normally think would be lethal to life. These particular organisms have adaptations that allow them to live at that elevated temperature. And as a matter of fact, they require higher temperatures to live. If you take an archaic thermophil and try to grow it at 37 degrees Celsius, it's not going to grow. It's adapted to living at that higher temperature. One of the interesting things is there are some of the adaptations that allow the enzymes of these bacteria to survive when they normally would expect them to denature at that particular high temperature. There's a very important thermophilic bacteria to the biotech industry called Thermophilus aquaticus, or TOC. Have you ever heard of TAQ before? TAQ? Anybody taken Dr. Morgan McComb's biotech class? There is a technique in biotechnology that allows us to amplify DNA called polymerase chain reaction. And polymerase chain reaction requires a heat-stable DNA polymerase, and the place we isolated originally from was these hot springs bacteria, this Thermophilus aquaticus bacteria. So that particular bacteria has been crucially important to the whole biotech industry. Anybody know what a halophil is? It loves something. It loves very salty environments, very hypertonic environments. What typically happens to a cell when you put it in a hypertonic environment? It shrivels, okay? And that actually is the basis of early attempts to preserve meat, for example, from bacterial contamination. You salted it, okay? You dehydrated it, okay? Well, the halophilic bacteria, like these guys right here, that are living in salt ponds, require high concentrations of solute in their environment to survive. They're adapted to those very saline type of environments. The Great Salt Lake, for example, is becoming increasingly saline over a period of time. And most fish can't tolerate it anymore. But the halophilic bacteria love it. The Dead Sea is very salty. And again, primarily bacteria that are halophilic are the major things that can survive in those very salty environments. Some of the halophilic bacteria produce some interesting pigments that allow them to convert sunlight into chemical energy. And these particular halophilic bacteria have some interesting colored pigments that allow them to do this kind of photosynthesis thing. We'll talk a little bit more about that later on. It's not technically photosynthesis. The pigment's called bacteria rhodopsin, but it's a way to convert light energy into ATP. Not all of the archaeans are extremophils. In fact, most of the archaeans aren't. They live in more or less normal type environments. One type of archaean that lives in a fairly normal environment is a group of microorganisms called methanogens. And these methanogens are metabolically able to combine carbon dioxide and water and form methane as a byproduct. So these are the methane producing bacteria. Have you ever heard that sometimes companies will try to harvest natural gas from a landfill where bacteria have been decomposing the stuff in the landfill? Okay. The natural gas that's produced is produced by these methanogenic archaean bacteria. Methanogens are oftentimes found buried under layers of mud in swampy land. Many of these are anaerobic bacteria, and being buried under those layers of mud prevents oxygen from penetrating into that environment and favors the growth of those methanogens. And as they metabolize waste material, decayed plant and animal material in that mud, they produce methane. And the methane can seep up out of the soil and into the water, and then come up to the top of the water in the form of bubbles that we call swamp gas. Okay? Methanogenic bacteria are also found in the gut of ruminants, cattle and sheep and things like that, that can eat cellulose because they have bacteria in their gut that can decompose them. Some of those are methanogens. And so ruminants oftentimes burp methane into the environment. Well, what's the significance of a lot of methane going into the environment from ruminants? In rice paddies, there are methanogenic bacteria that release CO or methane into the atmosphere. All of this methane in the atmosphere can do what?
destroy the ozone?
Doesn't destroy ozone. Methane is a gas that traps heat and contributes to global warming. Okay. Now, a certain amount of methane in the environment is actually beneficial, because without that methane and carbon dioxide in our atmosphere, the temperature of our planet would be very cold and very inhospitable. So a certain amount of heat being trapped contributes to global habitability. The problem is when methane levels and CO2 levels start increasing and we start getting too warm of an atmosphere, leading to things like global climate change. Okay? So ecologically, evolutionarily, these archaean bacteria have had a real impact in terms of kind of life on Earth. So these are some of the archaean bacteria and some of the classes that they're divided into. And again, you can kind of see some terms here that might now make a little bit more sense. Methano, okay, methane producers. Thermophilic organisms. Another methano right here. Halo, okay. So a lot of these archaeans are in those groups that I've just briefly mentioned. Okay? Within volume 1, we now also have some of the more traditional bacteria, members of the domain bacteria. And again, I don't want you to memorize all of the names of these particular things. I do want to point out a couple of them to you because we're going to encounter them later on. And that's going to be these last two phyla here. The phylocyanobacteria and the phylochloridia. So let's take a look at those for just a second. Cyanobacteria we've known about for a long time. And as a matter of fact, cyanobacteria used to be referred to as blue-green algae. And I think some of you probably, when you took botany, looked at some cyanobacteria in lab when you were studying algae. But these are bacteria. They have a peptidoglycan cell wall. They're gram-negative in nature. But the thing that's really important for us right now is that these cyanobacteria are photosynthetic, and particularly they carry out a type of photosynthesis that you should be familiar with, and that is oxygenic photosynthesis. The kind of stuff we talked about in cell biology, might have even talked about it in botany a little bit. So remember what happens in oxygenic photosynthesis. Water is split, and oxygen is released as a byproduct. You need some light energy to split that water, and that light energy is absorbed by a pigment, a type of chlorophyll typically. Okay. Light energy is responsible for initiating a hydrogen ion gradient. And the hydrogen ion gradient is used to make ATP. When water is split, the hydrogen is used to reduce NADP. And if we have some CO2 from the atmosphere, some ATP that you produced from your hydrogen ion gradient, and some reduced NADP, what can you make? You can make some glucose. It's a 90-second review of photosynthesis. Do you remember what this process is called, where the CO2 is reduced using ATP to make glucose? Remember what that cycle is called? Remember the Calvin cycle? That is the light-independent reactions of photosynthesis. These are the light-dependent reactions of photosynthesis. Coming back a little bit. Okay. So that's our typical oxygenic photosynthesis. And this is what cyanobacteria do. Okay. They have a little bit different type of photosynthetic pigment that's a bluish green color rather than the typical green color of chlorophyll. And that's why we call them that cyanobacteria. Cyan is blue. Okay. These particular organisms are primarily aquatic, although you can find them in terrestrial environments. One of the places that you can find cyanobacteria is in a symbiotic relationship with a fungus. And sometimes this symbiotic relationship of fungi and cyanobacteria can grow on rocks and the bark of trees. What do we call those symbionts? Heard of lichens before? Okay, so lichens are a symbiotic association of these cyanobacteria and fungi. One of the things that cyanobacteria is also important is that they are capable of carrying out a process called nitrogen fixation, which we mentioned already. But they do so in a non-symbiotic fashion. Remember the nitrogen fixation we mentioned at the beginning of the course? The bacteria that live in the roots of the legumes carry out symbiotic nitrogen fixation. And these non-symbiotic nitrogen fixers add a significant amount of usable nitrogen to the ecosystem. The cyanobacteria used to be one of the more dominant forms of life on our planet, particularly during the early stages of evolution of photosynthesis. These were really the first photosynthetic organisms that dominated the Earth. This is in a bay in New Zealand, and at low tide, you can see all of these pillars of green slimy stuff. Those green slimy pillars are called stromatolites, and basically they are large biofilms, primarily of cyanobacteria. This is what the world used to look like, a couple billion years ago, as it was dominated by these first photosynthetic organisms. And these organisms literally changed the planet by adding oxygen to the atmosphere, one bubble at a time, as a result of this oxygenic photosynthesis process. So the atmosphere became an oxidizing atmosphere. We also then later had an ozone layer which developed from the oxygen, which fundamentally changed the nature of the planet. So these cyanobacteria, evolutionarily, have been really important. Okay, have time. So don't forget to come in sometime, flip on the spec and get your readings, and then make your slant of your unknown. I've got to go to a thing at 5:30, so I won't walk in before that. Okay. Yes.
Turns out they figured that out.
so they know the flora of the space station.
Yeah,
The
yeah.
physical space station.
And they said the dominant species are in a, I guess it's a family called Acinetobacteria.
Mm-hmm.
So essentially, every astronaut that goes up there leaves a microbial trail.
Yes.
Yep.
Yeah.
But what they're concerned about is somebody doing something that will endanger the health of somebody there or somebody who comes later.
Mm-hmm.
But all they can do right now is just survey.
Yeah.
My guess is that the medical kits up there are full of antibiotics.